Hi everyone. So in the last video we talked about taking your RNA-seq reads and trimming them and then rechecking the quality of what those reads look like. Once you've done that, you're all good and clear to go towards alignment, which is often also called mapping. So you might see it called that as well. And this is where we are going to be matching up our reads, which are these kind of short fragments of our RNA transcript, to a reference genome. Which, just to note, should also be in this FASTA text file format. It'll probably just be these really long stretches of sequences. Sometimes it's even just like chromosome 1 and then the entire sequence of chromosome 1 and so on and so forth. So that'll also be a FASTA file. And I can link some resources for where you can get reference genomes if you don't have one already available to you, or you might have one kind of that your lab has generated, etc. But we're kind of matching these up. And the idea of that is let's say I have my reference genome up here, and then I have a bunch of different, it's not a very good color, a bunch of different reads that I'm going to match up to their respective locations on my reference genome. So these kind of all line up here. These ones line up here, so on and so forth. So we just want to understand, where do our reads fall on our reference genome, so that ideally in the next step of assembly, we can take this and say, okay, we have like full coverage. We have a read that matches to every single segment along this whole stretch. So maybe that is a whole stretch of RNA that's transcribed, right? And maybe certain portions of it are transcribed more highly, other portions of it are transcribed a little bit less. And then maybe we have a break, and then we have another set of reads that matches up right here. So we say, oh, there's another segment. And then we can use some type of gene prediction programs to say, is this part of the same gene or is this separate, right? But the alignment step is just taking each of our individual reads and matching them to where did they fall on this reference genome, right? And we're not actually assembling like a cohesive transcript. This would be assembly, which we'll talk about either later in this video or in the next video. But I'll erase this for now just so it's not confusing in terms of what we're doing with alignment. So I saw two main programs when I was looking into alignment. I saw HiSat2 and I saw RNAstar. And both of these programs I saw, kind of good reviews. Sounds so funny. But I saw kind of good reviews for them on bioinformatics forums in terms of the quality of their alignment. I think the big difference that you want to look at for your purposes is HiSat2 is fast, but maybe not as accurate. And RNAstar is slow, but highly accurate. So if you have a larger genome or some type of compromise in terms of your availability of RAM or other kind of computational requirements, HiSat2 might be a better option for you. Also, if your RNA-seq reads are of higher quality, HiSat2 might be a better option for you, because it's definitely not inaccurate. It's just not as good at sussing out the position of maybe hard to place reads, whereas RNA star is good if you have maybe less quality reads, or you have kind of maybe more of those computational resources, a smaller genome to work with. All of those things are going to make it a little bit easier to use RNA star. I actually personally used HiSat2 to start, and I figured if I had issues with assembly, I would go back and I would use RNA star. I didn't end up having really that many issues or issues with the quality of my alignment, so I just stuck with my HiSat2 data. So I think that's a totally fine way to go as well. I didn't really get any kind of warnings from people about that. The bioinformatics people I talked to said, yeah, that's totally fine. If it works, it works. So I ended up using HiSat2. And what you're going to end up with at the end of your alignment, we're finally kind of switching file types over to a BAM/SAM file. So these two files essentially contain the same information, except a BAM file is a more compressed version, and our SAM file is a less compressed version. So a SAM file is readable to the human eye. It has a bunch of different information that, again, I can make a video where I more thoroughly talk through all of what's included in a BAM file. We're just going to talk about kind of the main thing to watch out for, which is the MAP-Q score. So I'll go over that in a minute. But a SAM file is readable, and a BAM file is not. A BAM file is like alien gibberish. So if you wanted to freak someone out, you could sit in the library and open up a BAM file and act like you're reading it. And yeah, it's really crazy to look at. I've had some times where I accidentally outputted a BAM file, and it was insane to believe that it contained the same information as a SAM. But I will say for further computational processes, you want to have the BAM file available. Often, I would say, I think HiSat2 actually outputs both. So they'll output a SAM file for you to view and then a BAM file for you to save and use for further notice. I don't know about RNA star, but it'll be pretty obvious to you if you open it and you can't read it, then it's a BAM file. If you open it and you can read it, it's a SAM file. So that is kind of my my litmus test for which file type it is. And this is a little different format than FASTA. It has quite a bit more information, but the main thing that I would focus on from this is called the MAP-Q score. And the MAP-Q score is essentially very similar to a Phred score, but it's, instead of pertaining to individual base pairs and the confidence of their read, it indicates the confidence of a match or an alignment. So a higher MAP-Q score indicates that the program is more confident that an alignment is correct. Maybe more of the base pairs match, maybe there were less secondary matches, so confounding matches. Regardless of the reason, a higher MAP-Q score is good. It means that your alignments are more likely to be accurate. So often what we have to do after we go through this alignment process, you'll use HiSat2 or RNA star, you'll align, you'll get this BAM/SAM output, you'll take that BAM file and you'll go through a process called filtering again, but this time it's a little different. You're going to use a different program called SAM Tools, which I know is a little contradictory, but you can use a BAM file in SAM Tools. You can also convert in between a BAM and a SAM, or yeah, a BAM and a SAM in SAM Tools. So it's a very useful suite of tools that helps you deal with both BAM and SAM files. And filtering can be done using SAM Tools dot view, and you'll set a map Q threshold. I set mine at anything below a map Q score of 30 being thrown out. Now, for the majority of my reads, it was either a map Q score of like 0 or 1, or I had a map Q score of like 60. So 30 maybe was a bit of an arbitrary threshold, but you can look at your own data and see, do you have a bit more of a gradient in terms of your mapping quality? I think 30 is pretty standard from what I had read. So it's definitely like a safe metric to use, but you could set it higher if you feel like you see more of a gradient and you want to kind of restrict to really high quality reads. You could set it lower if you're feeling like, well, that's just going to cut out like all of my data. It's definitely a bit of an independent process to decide that map quality score. I will say the SAM Tools view process is very fast. So there's no problem to just run it again. It's not something where you're going to run it for 3 days and then not want to deal with running it again. It took like seconds on my MacBook Pro. So you'll be totally fine to run it as many times as you need to. If you find out that you need to kind of go back and restart, it's not a big problem, I would say. But you do need to filter out those low quality map scores of 0 and 1, etc., because they can cause problems in the next steps of your analysis. Because if they're included, then they can be factored in. So you want to make sure that you get rid of them using that SAM Tools view. Those really low quality maps do need to be gotten rid of. There's also a couple other things that we can do with SAM Tools that I'll list. So these kind of steps don't need to be done in any particular order, but I would highly recommend doing them once you have this HiSat2 output. Is you want to check your quality again, as always. Now, the map Q score is kind of one quality check that you can do, though I will say you're not going to scroll through all of the map Q scores in every single file. But the other quality check that you can do is called SAM Tools flag stat. And you can just input your BAM file, and I think that's literally the only input that you need for this. And you can look at kind of how many of the reads that were inputted were actually successfully mapped. And obviously, you want to see that there's kind of a high-ish proportion of reads that were successfully mapped. That indicates that your RNA-seq data was of a high quality and was kind of being successfully mapped to the reference genome. For my data, I was sitting at kind of 85 plus, which I've seen is kind of a good quality to be sitting at. If you had something like 30 or 40% mapping, you might want to go back and say, do I realistically have enough alignments to do something with this data? So it has a really nice, just easy output in the command line that you can look at. But I'll also, again, link to some graphical user interfaces that people can use for this as well. So a quality check is great to do. And then you can also use SAM Tools sort. And this is to sort all of your reads by the coordinates. So if you have an option of what you want to sort it by, you need to sort it by coordinates. This is necessary for the next step of assembly, so that your reads are sorted and not in like a random order. Some programs, to my understanding, do output a pre-sorted BAM file. Like I think that HiSat2 had already sorted my data based on the coordinates. And you can actually, when you look at the top of a HiSat2 file, it'll usually say like SO coordinates. And that indicates that it was actually already sorted by coordinates. But I'm not so sure about RNA star. So it never hurts to just run it. Again, it's a very, very quick process. All of the SAM Tools programs are taking less than 5 minutes on my MacBook. So especially if you have a more high-powered computer to run it on, you'll be totally fine to just run the sort and confirm that it was sorted by the coordinates. So those are kind of the three big things that you have to do with your HiStat2 or whatever aligner that you used. That output is you filter by the map quality score. You check your quality to make sure that a good number of your reads did align to your reference genome. And then you're going to sort by coordinates if that is not already the case in your BAM file. Now, if you've been working with a SAM file this entire time, again, that's the kind of human readable file, which is totally fine to work with so that you can kind of see the outputs a little more clearly. The last step you would need to do is convert to a BAM file. And this would be using SAM Tools View. You can choose your file output there, and you would just choose a BAM file as your output. Again, we can go through the code for that really easily if people are interested in seeing that. But yeah, that's kind of the process of alignment and what you have to do to your data after you align it. In the next video, I will talk about using StringTie, which is an assembler, my experience with that, and kind of processing the data afterwards to view it a little bit. All right, thanks for watching, y'all.